Here is Dr. Malik Nawaz from Department of Microbiology, Guwahati University of Science and Technology. Today my lecture number 13, which is on instrumentation and analytical technique. Today our lecture is in continuation with previous lecture based on different types of ELISA. These are the contents of my today lecture. So ELISA can be categorized into two groups on the basis of detection and on the basis of procedure. So on the basis of detection, there are three different types of ELISA. One is colorimetric ELISA, second one is the chemiluminescent ELISA, and the last one is the competitive ELISA. On the other hand, ELISA on the basis of procedure, we have non-competent ELISA, competent, competitive ELISA, multiple and portable ELISA, modified ELISA, and the last one is the modern ELISA. So each type of ELISA will be discussed in the coming slides. So on the basis of detection, we have colorimetric ELISA. The word colorimetric means color. The final product or the end product of ELISA which produce a color. So that the presence of the color indicates the positive of ELISA. So colorimetric ELISA basically used to find out or determine the concentration of antibody in a given sample. So this is a basically result in a color reaction product that absorbs light in the visible range. The optical density or the OD of the reaction product is typically proportional to the amount of analyte being measured in the sample. So initially, we coated the microtiter plate with the antigen in order to immobilize the antigen. Then we apply the primary antibody or capture antibody, which are monoclonal and they are specifically produced against this specific type of antigen. Then we apply the secondary antibody, which are modified, or in other words, we can say that these antibodies are linked with the enzyme. And we in three types of enzyme has been reported which are attached with the secondary antibody. It may be attached with the primary antibody in case of direct ELISA, in which only one type of antibody is used. So the label antibody, the attachment of the enzymes may be horseradish peroxidase, it may be alkaline phosphatase, or it may be beta galactosidase. So any type of enzyme can be linked with the secondary antibody. And the last one, a substrate is added to the reaction and the enzyme convert the substrate into the final chromogenic product, which can be further used or read in the ELISA later. So this is the colorimetric ELISA. The second type of on the basis of detection, we have the chemiluminescent ELISA. So this type of ELISA basically is used for the quantification of an antigen in a given sample. In this type of ELISA, antibody coated on the plate which specifically bind to the desired analyte. Following a washing steps, a detection antibody and enzyme label are added which bind to the analyte. A substrate is finally added to generate a reaction in the wells. Depending on the type of assay, either color or light produced. So in this case, this reaction can be measured by plate reader. The degree of light emitted indicates the amount of antigen or analyte in this sample. This elucidates the direct proportionality of a signal strength to concentration of antigen present in the biological sample. Here we can see that the primary antibody or capture antibody loaded on the microtiter plate in order to make them immobilized. Then antigens are applied. So these antigens are bound to these monoclonal capture antibodies. Now, addition of the secondary antibody, which are enzyme links, I have already discussed. These different types of enzyme which are linked with the are coded on the secondary antibodies, and addition of substrate which are produce a specific links. So this indicates that when there is a complex, there will be emission of light. When there is no complex, there will be no emission of light. So emission of light directly proportional to the amount of analyte present in the given sample. So these are increasing or decreasing in direct proportionality. So the third type on the basis of detection is the competitive ELISA. The word competitive indicates a competition between two things, A and B. So A may be a simple, so this competition may be between two types of antibodies, which is one is simple antibody, the other is conjugated antibody or maybe more modified antibody. It may be two types of different antigens. One may be specific antigen, the other may be alternative antigens. So it basically used for a competition in order to find out the concentration of antibody or antigen in a given sample. For example, the a competitive binding assay is based upon the competition of a labeled and unlabeled reagent for a limited number of antibody binding sites. Only one antibody is used in competitive binding ELISA. So competitive binding assays are often used to measure small analyte. This ELISA is predominantly used to measure the concentration of an antigen or an antibody in a sample by detecting interference in an expected signal out. If antigens are more in reaction, then signal strength will be reduced. So this indicates irreversible proportionality or indirect, they independent on each other. Suppose that the microtiter plates wells are coated with the capture antibody, then specific antigens are applied. So we have two types of antigens. So one is the addition of antigen and the other one is the antigen conjugated enzyme complex. Here we can see. So both are applied and they are equally fighting for the are competent for to bind with the antibody. So when the when they are attached to the antibody, so they produce a signals, the final products, which can be measured. So when the number of antigens are more, then the absorption will be less. When the absorption is more, this indicates the less number of antigens or analog of antigens are present in the given sample. So this clearly indicates the inverse or indirect proportional to one another. So this is a type of competition between two different things. On the basis of procedure, we have a non-competitive ELISA. So there are three different forms, different subtypes of non-competitive ELISA. One is the direct ELISA. In direct ELISA, there is only one type of antibody is used, and this capture antibody or this monoclonal antibody is usually linked with an enzymes. So the wells are coated with the specific antigens and then followed by the uh, capture antibody which are linked with an enzyme when substrates are applied. So this enzyme basically converts the substrate into the final chromogenic product and we are getting the results. In the indirect ELISA, in this type of ELISA, wells are coated with the antigens, then we followed by the capture antibody. In the capture antibody, they are produced against this specific type of antigens. So for the recognition of primary antibody, we are using the secondary antibody, which is a modified antibody and linked with the N enzymes. After the addition of substrate to the reactions, so the enzyme convert the substrate into the final product, and the OD may be measured using the ELISA plate reader. So in the indirect ELISA, we can say that we are using two different antibodies. One is the capture or primary antibody, and the second one is the secondary antibody. On the other hand, we can use sandwich ELISA. So the word sandwich indicates two types of antibodies which sandwich the antigen. It can be simply explained that it is used for determining an antigen in a given sample. Antigen in this sample binds with the capture antibody and become immobilized. The antibody of the enzyme conjugate bind with the immobilized antigen to form a sandwich of antibody, antigen, antibody, enzyme bound to microtiter well. Microtiter wells are initially coated with monoclonal antibodies called capture antibody raised against antigen to be detected, finally followed by the addition of a sample. So any trace of antigen is detected by adding primary antibody, which are usually monoclonal antibody, followed by enzyme conjugated secondary antibody and a chromogenic substrate, or by directly adding an enzyme conjugated primary antibody. Here we can see that wells are coated with the capture antibody or monoclonal antibody. So when antigens are applied, these antigens are uh, specific to these monoclonal antibody, so they make a complex. Now we have another antibody which is known as secondary antibody. When we apply the secondary antibody, so it bind here. Secondary antibody are linked with another enzymes. So here we have biotin and biotin which are specifically vitamin vitamin vitamins, water soluble vitamins, and also known as vitamin H. For more specificity, another protein product, avian, which are present in the reptiles and birds, and they are basically producing a complex protein binding proteins, and they are linked with the horseradish peroxidase enzyme. After the addition of substrates, the, the enzyme convert the TMB into chromogenic product. So here we can see the antigen is sandwiched between primary antibody and secondary antibody. Competitive ELISA I have already explained in detail, which can be used to find out the concentration of antigen or concentration of antibody in a given sample. This slide indicates the comparison of indirect ELISA sandwich and competitive ELISA. I have explained each and every type of ELISA in detail. In multiple and portable ELISA, this is a newer class or a modified version of ELISA. This tool uses an solid phase made up of an amino-sorbent polystyrene rod having 8 to 12 protruding ogives. So here we can see it may be 8 or it may be 12, and they are all present in the form of a comb, in which the teeth are protruding teeth may be present. So this is known as ogives. So the entire device is immersed in the test tube containing the collected sample, and the following steps such as washing, incubation in conjugate and conjugation and incubation in homogeneous material are carried out by dipping the ogives in micro-titer wells of standard microplates pre-filled with reagents. So this ELISA is applicable in human health, food safety, animal health, agriculture, and water, and so many other application has been reported. Modified ELISA. This is also a type of ELISA in which we have done some sort of modification. So in modified ELISA, microtiter wells are coated with the capture antibody followed by the addition of antigens. So these antigens are specific to these monoclonal antibodies because this monoclonal antibody has been produced against these specific antigens. So they are making a complex. So this complex gives another antibody which is known as secondary antibody, which are linked with biotin. Biotin is a soluble, water soluble vitamins known as vitamin H, and that's a part of vitamin B and currently known as vitamin H. So this antibody also making a complex. So now the antigen is sandwiched between the primary antibody and the secondary antibody. So biotin is basically making a complex with avidin, which is linked with the another enzymes known as horseradish peroxidase. So when they are making a complex, then another uh, chromogenic substrates are added, which convert the substrate into the chromogenic product, a readable product. So this type of ELISA has more sensitivity and specificity as compared to the rest of all. And